Good afternoon. In this T7 presentation, I'll be discussing meiosis and the differences between meiosis and mitosis. Here is the YouTube link to my video that you're watching right now. Meiosis is a source of distinction. Why do you share some but not all characteristics of each parent? What are the rules of this sharing game? At one level, the answer lies in meiosis. Meiosis does two things. First of all, meiosis takes a cell with two copies of every chromosome, a diploid, and makes cells with a single copy of every chromosome, a haploid. This is a good idea if you're going to combine two cells to make a new organism. This trick is accomplished by halving the chromosome number. In meiosis, one diploid cell produces 4 haploid daughter cells. Second, meiosis scrambles the specific forms of each gene that each sex cell, sperm, or egg receives. This makes for a lot of genetic diversity. This trick is accomplished through independent assortment and crossing over. Genetic diversity is important for the evolution of population and species. Why do we need meiosis? Well, meiosis is necessary to have the number of chromosomes going into the sex cells. But why do we need to have the chromosomes and gametes in the first place? Well, at fertilization, the male and female sex cells will provide 1/2 of the chromosomes each, resulting in offspring that have genes from both parents. The parent cell has a chromosome pair. The chromosomes are then copied. The first division, the pairs split, and in the second division, produces 4 gamete daughter cells with 1/2 the original number of chromosomes, as shown in this graphic. Meiosis is also known as a reductive division because we're reducing the number of chromosomes from 2 to 1. The stages of meiosis in a nutshell. Meiosis 1 is where homologous chromosomes are separated. In interphase, prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis. And we'll go further in depth into these different stages as we progress through the slideshow. In interphase, the chromosomes duplicate. In prophase, the non-homologous or the homologous chromosomes pair and exchange segments. In metaphase 1, the tetrads line up. And in anaphase, the pairs of homologous chromosomes split up. Meiosis 2 is where the sister chromatids are separated, and it proceeds similar to mitosis. There is one important key note here. There is no interphase 2 because there's no further replication of the DNA. Meiosis 2 consists of prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. During telophase 1 and cytokinesis, a cleavage furrow forms, and 2 haploid cells form. Chromosomes are still double. During another round of cell division, the sister chromatids finally separate, and 4 haploid daughter cells are the result, containing single chromosomes, as shown in this graphic, outlining meiosis 2. And we'll get further into the details of meiosis 2 in future slides. Meiosis 1 in detail. Interphase. Each of the chromosomes replicate. The result is 2 genetically identical sister chromatids which remain attached at their centromeres. Prophase 1. This is a crucial phase for mitosis. The nuclear envelope breaks down and homologous chromosomes pair out. During this phase, each pair of chromatids don't move to the equator alone. They match up with their homologous pair and fasten together in a synapsis in a group of 4 called a tetrad. It is extremely important to note that this phase is when crossing over can occur. Crossing over is the exchange of segments during synapsis. Crossing over can occur or occurs when portions of a chromatid on one homologous chromosome is broken and exchanged with the corresponding chromatid portions of the other homologous chromosome. Metaphase 1. Homologous pairs of chromosomes line up at the equator attached by the centromeres to special to spindle fibers from centrioles. Anaphase 1. The spindle guides the movement of the chromosomes toward the pole. Sister chromatids remain attached. They move as a unit toward the same pole. The homologous chromosome moves toward the opposite pole. This is different than mitosis, where chromosomes appear as individuals instead of pairs. Telophase 1. This is the end of the first meiotic cell division. The cytoplasm divides, forming two new daughter cells. Each of the newly formed daughter cells has half the number of the parent cell's chromosomes, but each chromosome is already replicated and ready for the second meiotic cell division. Cytokinesis. This occurs simultaneously with telophase 1. It results in the formation of two daughter cells. Plant cells form a cell plate, while animal cells form cleavage furrows. There is no further replication of genetic material prior to the second division of meiosis. Meiosis 2. This phase is responsible for the separation of sister chromatids. It proceeds similar to mitosis. There is no interphase 2. There is no interphase 2 because there's no need to duplicate or replicate the chromosomes. Prophase 2. Each of the daughter cells form a spindle. The double-stranded chromosomes move toward the equator. Metaphase 2. The chromosomes gain position along the metaphase plate in a mitosis-like manner. They are attached at centromeres by spindle fibers. Anaphase 2. The centromeres or sister chromatids finally separate. The sister chromatids of each pair move toward opposite poles as individual chromosomes. Telophase 2 and cytokinesis. Nuclei form at the opposite poles of the cell and form around each set of chromosomes. Cytokinesis then occurs, resulting in 4 haploid, or N, daughter cells. So how does mitosis or meiosis create such a variation? One way is through independent assortment. Independent assortment produces 2 to the nth power of distinct gametes, where n equals the number of unique chromosomes. In humans, n equals 23, so that would be 2 to the 23rd power, which is a huge number, over 6 million. That is a lot of diversity by this mechanism alone. This graphic here shows independent assortment of maternal and paternal homologs during meiotic division 1 through meiotic division 2, and the possible gamete outcomes. Meiosis making even more different sex cells through crossing over multiplies the already huge number of different gamete types produced by independent assortment. This happens because of the exchange of parts of non-sister chromatids. The duplicated maternal chromosome and the duplicated paternal chromosome come together to form a tetrad. They are non-sister chromatids, the paternal and the maternal. They come together, form a tetrad, and then independent assortment occurs, and they come apart, and as this graphic shows, different portions stick with different chromatids. Key differences between mitosis and meiosis are in the way the chromosomes uniquely pair and align in meiosis. In mitosis, the chromosome pairs, homologous pairs, form along the pole and are pulled, or along the equator and are pulled along to the poles by the spindle fibers. During the first and most distinguishing division of meiosis, the sister chromatids are pulled together. Mitosis versus meiosis. A summary. DNA replication in mitosis occurs during interphase before nuclear division begins. DNA replication during meiosis occurs once during the interphase before meiosis 1 begins. The number of divisions in mitosis is 1, including prophase, metaphase, anaphase, and telophase. The number of divisions in meiosis is 2, each including prophase, metaphase, anaphase, and telophase. The synapsis of homologous chromosomes does not occur in mitosis. Synapsis is unique to meiosis. During prophase 1, the homologous chromosomes join along their length, forming tetrads, or groups of 4 chromatids. Synapsis is associated with crossing over between non-sister chromatids. Number of daughter cells and genetic composition in mitosis is 2, each diploid 2N and genetically identical to the parent cell. The number of daughter cells and genetic composition in meiosis is 4, each haploid or N containing half as many chromosomes as the parent cell, genetically non-identical to the parent cell or to each other. The role in the animal body of mitosis is that it enables multicellular adult to arise from a zygote. It produces cells for growth and tissue repair. And the role in the body for meiosis is that it produces gametes. It reduces chromosome number by half and introduces genetic variability among the gametes. Crossing over. The DNA exchange that occurs during crossing over adds even more recombination to the independent assortment of chromosomes that occurs later in meiosis. Therefore, the number of genetic combinations that can occur among gametes is practically unlimited. The zygote that forms a new individual is created by the random joining of 2 gametes. The importance of variation in genetics. Meiosis and the joining of gametes are essential to evolution. No genetic process generates variation more quickly. The pace of evolution is sped up by genetic recombination. The combination of genes from 2 organisms results in a third type not identical to either parent. Meiosis in males. This differs from meiosis in females in one key way, and we'll get into that right now. The process by which a sperm is produced in a male animal is called spermatogenesis. This occurs in the testes, which are the male reproductive organs. This produces male gametes called sperm. During sperm formation, cytoplasm is divided equally after the first meiotic division. Then, the cytoplasm is again divided equally after the second meiotic division. This results in 4 equally sized viable sperm cells from each cell that undergoes meiosis. Meiosis in females. This is a process by which gametes are produced by females, and it is called oogenesis. Oogenesis occurs in the ovaries, or the female reproductive organs. It produces a female gamete called an ova or an egg. During egg formation, the cytoplasm is divided unequally after both meiotic divisions. This results in 1 large egg or ovum cell and 3 smaller ones. The 3 small ones subsequently die. Thus, only 1 egg is created from each parent cell through meiosis. Important to note, male meiosis results in 4. Female results in 1 for gametes. And that is because the 3 smaller gametes that are produced by the eggs, meiosis, end up dying because they're not sustainable. Comparing some differences between mitosis and meiosis. In meiosis, the chromosomes line up differently. Homologous chromosomes pair up to form tetrads. Crossing over occurs, resulting chromosomes are different from the parent cell. In mitosis, the chromosomes line up singly with no crossing over occurring. The separation of chromosomes. Chromatids are still attached to each other as the homologous chromosomes separate in meiosis. In mitosis, homologous chromosomes line up singly and the chromatids separate. The result of division. In meiosis, each cell produced has only 1 chromosome out of each pair of homologous chromosomes. The result of division in mitosis, each cell produced has 1 copy of both of the homologous chromosomes. Mitosis stops here. The second division. Daughter cells divide again in meiosis. Chromatids separate. Now results in 4 daughter cells with only 1 copy of every pair of homologous chromosome. In mitosis, there is no second division. So as we've covered and discussed, there are many key differences between meiosis and mitosis. The goal of meiosis is to take one parent cell and turn it into 4 daughter cells containing half of the nuclear information or the DNA information to be passed on to form a zygote. Here are my references. Feel free to click the links if you would like to read the articles. And I thank you for your time, and I hope that you learned a lot from my presentation.